students, and welcome to the lecture on signaling molecules and their receptors, function of cell surface receptors, intracellular signal. The main objectives of today's lecture are to give a brief introduction of cell signaling, to explain signaling molecules, to give a detailed account of the receptors of various signaling molecules, to illustrate various functions of cell surface receptors, to give the pathways followed in intracellular signaling. To begin with, let me first of all give a brief introduction about cell signaling. One characteristic common to all organisms is the dynamic ability to coordinate constantly their activities with environmental changes. Cell signaling makes it possible for cells to respond in an appropriate manner to a specific environmental stimulus. This is accomplished by a variety of signaling molecules that are secreted or expressed on the surface of one cell and bind to receptors expressed by other cell, thereby integrating and coordinating the functions of many individual cells that make up organisms. Extracellular signaling usually involves the following steps. Number 1, synthesis and release of the signaling molecules by the signal cells. Number 2, transport of the signal by a specific receptor leading to its activation. Number 3, initiation of signal transduction pathways. In animals, extracellular signaling by signaling molecules can be classified into 4 categories: autocrine, paracrine, endocrine, and juxtacrine signaling. Now, let me begin with the first part of today's lecture, that is the signaling molecules and their receptors. Signaling molecules are the molecules that are responsible for transmitting information between cells in the body. The size, shape, and function of different types of signaling molecules can vary greatly. Signaling molecules fall into a variety of different chemical classes. Since the vast majority of signaling molecules in a chemical class do have the same polarity, let us discuss the signaling molecules based on their polarity. They are of 2 types: hydrophilic signaling molecules and hydrophobic signaling molecules. Hydrophilic signaling molecules. Water-soluble signal molecules are polar and therefore cannot pass through the nonpolar center of the plasma membrane unaided. Instead, most water-soluble ligands bind to the extracellular domain of cell surface receptors. The majority of the hydrophilic signaling molecules are based on amino acids. They may be unmodified amino acids, modified amino acids, or strings of amino acids, that is peptides or proteins. Some additional small polar signal molecules also are hydrophilic. Now, let me give you a brief description of the hydrophilic signaling molecules. Number 1, amino acids. There are 4 amino acids that function as signal molecules. All 4 are neurotransmitters that are synthesized and stored in vesicles by the neuron that release them. They are glutamate, aspartate, glycine, and gamma-aminobutyric acid. Number 2 is the biogenic amines. All the members of this group are hydrophilic except thyroid hormone. The biogenic amine molecules all contain an amine group and are derived by modifying an amino acid. This group can be further differentiated by the specific amino acid that is modified to make the signal molecules. They include catecholamines, thyroid hormone, histamine, and serotonin. All are synthesized in the cytosol of the cells that release them and are stored in vesicles. Thyroid hormone. Thyroid hormone is derived from 2 tyrosine molecules. Thyroid hormone molecules differ from others in the class by being hydrophobic rather than hydrophilic, and are like most other hydrophobic signals, its mode of action is intracellular. Even though thyroid hormone is hydrophobic and has intracellular receptors, the molecules do not passively diffuse across the plasma membrane but must be actively transported across to reach their receptors. Thyroid hormone can act on almost all cells in the body and is involved in setting the metabolic rate and in cell growth and division. Histamine. Histamine is derived from the amino acid histidine. It is best known for its paracrine signaling function in the immune response, which causes inflammation and the symptoms of many allergies. However, it also is an important signal molecule involved in the gastrointestinal signaling and as a neurotransmitter. In the GI tract, histamine acts as an enteroendocrine hormone that stimulates the release of gastric acid. As a neurotransmitter, histamine release from the hypothalamus increases wakefulness and prevents sleep. Serotonin. Serotonin is derived from the amino acid tryptophan. Serotonin is involved in a variety of important functions. In the GI tract and blood platelets, the general function is to cause smooth muscle contraction. However, the most publicized function of serotonin is its association with general mood and mood disorders in the central nervous system. The most well-known are the selective serotonin reuptake inhibitors that block the reuptake channels in the presynaptic cells, thereby increasing the time and concentration of serotonin in the synapse. Peptides and proteins. The majority of the signaling molecules are peptides and small proteins. All interact with extracellular cell membrane receptor proteins. The peptides and proteins are synthesized as inactive large molecules called prepropeptides in the rough ER of the cell that release them. The final version of the propeptide is stored in the vesicle and released by exocytosis. After hydrophilic signaling molecules, let me now explain some hydrophobic signaling molecules. Small hydrophobic ligands can directly diffuse through the plasma membrane and interact with internal receptors. Two important members of this class of ligands are the steroid hormones and eicosanoids. Other hydrophobic hormones include thyroid hormones and vitamin D. In order to be soluble in blood, hydrophobic ligands must bind to hydrophilic carrier proteins while they are being transported through the bloodstream. Now, let us discuss few of them. Steroids. Steroids are lipids that have a hydrocarbon skeleton with 4 fused rings. Different steroids have different functional groups attached to the carbon skeleton. Cholesterol is the precursor of the steroid hormones. Steroid hormones include estradiol, testosterone, cortisol. Since these molecules are small and hydrophobic, they can diffuse directly across the plasma membrane into the cell, where they can interact with the internal receptors, although some also bind to cell surface receptors. Eicosanoids. Eicosanoids are also synthesized from arachidonic acid found in the plasma membrane. Eicosanoids begin as the plasma membrane phospholipids and that are modified by 2 different pathways. The cyclooxygenase pathway creates prostaglandins, prostacyclins, and thromboxanes. The lipooxygenase pathway creates leukotrienes. In addition to these signaling molecules, other important signaling molecules include calcium ions, nitric oxide, non-coding RNA. After discussing the endogenous signaling molecules, here it's mandatory to give you very brief information of exogenous signaling molecules. When the signal molecule is produced by our body, we call it an endogenous signal. When the signal molecule is produced by an external organism or artificially, we call it an exogenous signal. Many receptors are named for the exogenous signals that were used to identify them. For example, nicotinic from tobacco, muscarinic from mushrooms, cannabinoid from cannabis. They all have endogenous counterparts. Exogenous signal molecules can be divided into 4 major sources: bacterial, multicellular tissue, pheromones, synthetic molecules. Now, let me start the second part of today's topic, that is the signaling molecules receptors. The cellular response to a particular extracellular signal molecule depends on its binding to a specific receptor located on the surface of a target cell or in its nucleus or cytosol. Receptors are chemically proteins or glycoprotein molecules which bind to a signaling molecule termed ligand. Binding of a ligand to a receptor causes a conformational change in the receptor that initiates a sequence of reactions leading to a specific cellular response. Based on location, receptors are classified into two broad categories: intracellular receptors and cell surface receptors. Intracellular receptors. The most prominent class of the intracellular or cytoplasmic receptors comprises the nuclear receptors that are found in the cytosol and or in the nucleus. Intracellular receptors include receptors for steroid hormones, thyroid hormones, retinoids and vitamin D, as well as different orphan receptors. The intracellular receptors are all structurally related and belong to the nuclear receptor superfamily. Most members of the nuclear receptor superfamily consists of an N-terminal activation domain, also known as AB region, a central DNA binding domain, and a C-terminal ligand binding domain. N-terminal domain contains a transcription activation domain AF1 and is recognized by coactivators and other transcription factors. The ligand binding domain also contributes to the homodimerization of many nuclear receptors. Cell surface receptors. These cell surface receptor proteins act as a signal transducers to produce intracellular messengers. Binding of ligand to some of these receptors induces second messenger formation, whereas ligand binding to other does not. For convenience, we can sort these receptors into four classes: G protein coupled receptors, ion channel receptors, tyrosine kinase linked receptor, and enzyme linked receptor. Now, let's discuss them briefly one by one. Number 1, G protein coupled receptors. Ligand binding activates a G protein, which in turn activates or inhibits an enzyme that generates a specific second messenger or modulates an ion channel, causing a change in membrane potential. The receptors for epinephrine, serotonin, and glucagon are examples. Number 2, ion channel receptors. Ligand binding changes the conformation of the receptor so that the specific ions flow through it. The resultant ion movements alter the electric potential across the cell membrane. The acetylcholine receptor at the nerve muscle junction is an example. Number 3, tyrosine kinase linked receptors. These receptors lack intrinsic catalytic activity, but ligand binding stimulates formation of a dimeric receptor, which then interacts with and activates one or more cytosolic protein tyrosine kinases. The receptors for many cytokines, the interferons, and human growth factor are of this type. These tyrosine kinase linked receptors sometimes are also referred to as cytokine receptor superfamily. Number 4, receptors with intrinsic enzymatic activity. Several types of receptors have intrinsic catalytic activity, which is activated by binding of ligand. For instance, some activated receptors catalyze conversion of guanosine triphosphate to cyclic guanosine monophosphate. Others act as a protein phosphatases, removing phosphate groups from phosphotyrosine residues in substrate proteins, thereby modifying their activity. The receptors for insulin and many growth factors are ligand-triggered protein kinases. In most cases, the ligand binds as a dimer, leading to the dimerization of the receptor and activation of its kinase activity. These receptors, often referred to as receptor serine threonine kinase or receptor tyrosine kinases, autophosphorylates residues in their own cytosolic domain and also can phosphorylate various substrate proteins. Our next subtopic is the function of cell surface receptors. Number 1, cell surface receptors are responsible for the binding of an extracellular signaling molecule and transduction of its messages into one or more intracellular signaling molecules. A cell surface receptor has two domains of significance: the signal molecule binding domain, which is exposed to the exterior of the cell, and the intracellular domain in contact with the cytoplasm. Therefore, it transmits external information to the interior of the cell in a regulated manner. Ligand-gated ion channel receptors are responsible for the rapid transmission of signals across synapses in the nervous system by changing the membrane potential. Ionotropic receptors transduce a chemical signal in the form of pulse of neurotransmitters delivered to the outside of the target cell directly into an electrical signal in the form of change in voltage across the target cell's membrane. Some neurotransmitter receptors directly control ion flux across the plasma membrane. Enzyme-linked receptors are typically single-pass transmembrane receptors with the enzymatic component of the receptors kept intracellular. The majority of the enzyme-linked receptors are associates with protein kinases. G-protein coupled receptors are integral membrane proteins that possess 7 transmembrane helices. These receptors activate a G-protein with ligand binding. G-protein is a trimeric protein. The 3 subunits are called alpha, beta, and gamma. The alpha subunit after activation dissociates from the beta and the gamma subunits. The activated alpha subunit can further affect intracellular signaling proteins or target functional proteins directly. In addition to regulating target enzymes, both alpha, beta, and gamma subunits of some G proteins directly regulate ion channels. The epidermal growth factor receptors function as a protein tyrosine kinase, establishing protein tyrosine phosphorylation as a key signaling mechanism in the response of cells to growth factor stimulation. The majority of the cell receptors are receptors for growth factors such as epidermal growth factor, platelet-derived growth factor, fibroblast growth factor, hepatocyte growth factor, nerve growth factor, and hormones such as insulin. Now, moving on to the last part of today's lecture, that is the intracellular signaling pathways. Molecular signals are transmitted between cells by the secretion of hormones and other chemical factors, which are then picked up by different cells. Sensory signals are also received from the environment in the form of light, taste, sound, smell, and touch. Once a receptor has received a signal, it must transmit this information effectively into the cell. This occurs via signal transduction pathways that are very specific in their activation and in their downstream actions. The intracellular component of signal propagation, also known as signal transduction, is receptor specific. A given receptor will activate only very specific sets of downstream signaling components, thereby maintaining the specificity of the incoming signal inside the cell. In addition, signal transduction pathways amplify the incoming signal by a signaling cascade, resulting in an appropriate physiological response by the cell. Several small molecules within the cell act as intracellular messengers. These molecules include cyclic adenosine monophosphate, cyclic guanosine monophosphate, nitric oxide, and calcium ions. Increased levels of calcium ions in the cell can trigger several changes, including activation of signaling pathways, changes in cell contraction and motility, or secretion of hormones or other factors depending on the cell type. Similarly, increased levels of nitric oxide cause relaxation of smooth muscle cells and vasodilation by increasing cyclic guanosine monophosphate levels within the cell. Increasing cyclic adenosine monophosphate levels can modulate signaling pathways by activating the enzyme protein kinase A. One of the most important functions of cell signaling is to control and maintain the normal physiological balance within the body. Activation of different signaling pathways leads to diverse physiological responses such as proliferation, death, differentiation, and metabolism. Signaling pathways in cells may also interact with each other and serve as signal integrators. Students, that's all about today's program. Hope you have understood and enjoyed well. Thank you and goodbye.